Hi, my name is Adel Far, and I manage technical development of drug substances at the medicines company, now part of Novartis Pharmaceuticals. I wanted to start my presentation by thanking Marc Lemaitre and the organizing committee of the OTS in giving me the opportunity to present some of our works. So the title of my presentation is The Control of GalNAc siRNA Conjugates. And as a disclaimer, I'm an employee of Novartis Pharmaceuticals. The experience that we've built with oligonucleotides is based on inclisiran sodium. The medicines company is not a traditional oligonucleotide company. We've developed drugs in the injectable market for a relatively long time, and inclisiran was really our introduction to oligonucleotides. It came about as a collaboration between the medicines company and Alnylam Pharmaceuticals. Alnylam discovered and developed the drug up to phase 2, and from phase 2 onwards up to filing, the medicines company was taking charge of this particular siRNA. The medicines company has been acquired now by Novartis Pharmaceuticals and is part of that organization. The structure of the drug is kind of schematically represented there. It's a small interfering double-stranded RNA. Nothing unusual there in the Alnylam stable of drugs. It has the triantennary GalNAc conjugate to allow it to be delivered to hepatocytes. And as you can see schematically there are two phosphorothioate at the end of the sense strand and two at each end of the anti-sense strand. Otherwise, the backbone is mostly phosphodiester. Inclisiran is meant to inhibit the production of PCSK9 in a hepatocyte. PCSK9 is a down regulator of the LDL receptor. By inhibiting PCSK9, we increase the LDL receptor on cell surfaces, and in turn, LDL receptor picks up LDL cholesterol from circulation. So it's a drug that's meant to treat hypercholesterolemia. It is a second generation siRNA from the Alnylam perspective, so it's not formulated in LNPs. And as most of these drugs from the Alnylam stable are, and as Chris presented in his ADME talk, inclisiran has a very long pharmacodynamic effect. The drug is out of circulation within 24 hours from plasma, but the pharmacodynamic effects last months. So in two phase 3 trials, inclisiran was compared to placebo, and in a very infrequent dosing regimen. The dose was subjected at day 0 at 3 months, and then 6 months after that. And over the duration of the treatment, the average level of cholesterol is decreased by 50% in patients. The patients that are getting placebo are receiving standard of care statins and other drugs that are meant to control their cholesterol levels. So there is a significant effect associated with inclisiran sodium, and it is hoped that inclisiran will have a major impact on cardiovascular disease. That's the clinical pitch, but in terms of my presentation, the focus of the talk is really on quality control of the drug substance. The inclisiran drug substance is manufactured through solid phase synthesis. Nothing unusual there from the perspective of oligonucleotides. There's a chromatographic purification step. The two single strands are isolated, and then they're annealed to form the duplex, which is then concentrated and lyophilized to provide the drug substance. We will not describe too much in detail about starting materials in this talk, and I'm going to mostly focus on the drug substance itself. In terms of control of the drug substance, we have the typical non-purity parameters associated here. So appearance, identity, microbiological attributes, moisture assay, these are typical analytical toolbox items. There are specific requirements associated with inclisiran and with oligonucleotides in general, in particular sodium content, pH in solution are things that regulators expect from us. One of the items that is relatively important is sequence confirmation. There are a number of ways to do this. Some companies do it by failure sequence analysis, others do it by MS fragmentation, which is what we're doing here. The sequence confirmation is performed on single strands. It is a relatively important item in the specifications of these drugs. Regulators have a true concern that in solid phase synthesis, the wrong phosphoramidite is used in the wrong sequence. And so you might end up with something with the same molecular weight, but a slightly different sequence. So it's something that they look relatively seriously on. In our case, there has been some discussion from regulators about trying to do this on the duplex. We've argued that it's too complicated to do it on the duplex, and once confirmed on the single strands, there's relatively little risk in ending up with the wrong sequence at the duplex stage, and that seems to be something that they've agreed on. In terms of purity, we apply the typical ICHQ3A approach and divide the impurities into two categories: process-related impurities and product-related impurities. Process-related impurities are impurities that are not structurally related to the main drug. Mostly these are reagents, solvents, byproducts that are generated during the manufacturing process. In this particular case, for inclisiran and for most oligonucleotides, the process-related impurities are really generated during the solid phase synthesis. There's relatively little generated downstream of that. And therefore, in our approach in this, we mostly use a risk analysis based on theoretical purge factors as suggested by Teasdale for mutagenic impurities. And so with the very large theoretical purge factors associated with having solid phase synthesis so far upstream from the final drug substance, there is relatively little risk of these materials finding themselves into the final drug substance. This includes mutagenic and non-mutagenic compounds, and we've used the same approach for both of them. With solvents, we've used a slightly different approach. Solvents were determined during development batches, and they're evaluated by risk analysis. And so we've used development work looking at the final drug substance to demonstrate that the large purge factors expected for solvents are justified. And that allows us to select which solvents are specified in the drug substance and which ones are not. We've used the same approach for elemental impurities. We evaluated the potential for elemental impurities as per ICHQ3D. And these elemental impurities were tested in development drug substance batches. And therefore a risk analysis based on actual data is used to evaluate these. One of the items that's recently added to the list of process-related impurities are nitrosamines. There is relatively little risk of nitrosamines present in oligonucleotides, and a risk analysis based on theoretical evaluation is what we've moved forward with. But with the two guidances, the EMEA guideline and now there is an FDA guidance as well on nitrosamines, it's something that you know, anyone developing this type of oligonucleotides has to remember to leave in their in their evaluations. Most of our purity then is really kind of associated with product-related impurities. These are oligonucleotides that are structurally related to the drug substance and to the single strands. Most of these product-related impurities are well described in the literature, and there's this very good kind of seminal paper by Capaldi and co-workers that has provided a good summary of these. Thomas went into greater detail about this, so I'm going to focus mostly on describing these. You know, the typical categories are well known: short-mered, long-mers, you know, PO impurities, processed side products, partially deprotected oligonucleotides, and degradation products. In our case, defluorination, depurination are particular concerns. There is a specific kind of category of impurities here, which are related to the GalNAc. Although I have to say, given the prominence of GalNAc oligonucleotides in the industry now, it's becoming a relatively common category of impurities. There should be an effort to identify these impurities, and simply relying on literature is not sufficient. The platform nature of oligonucleotides manufacturing obviously makes that easier. The purities are well known in the literature, and so in many cases, they can be identified by simple techniques such as LCMS, and simply compared to theoretical and molecular weight. There are relatively few impurities that are specific to this particular drug substance, and so there has to be a discussion as to how much identification is needed here and how much characterization is needed. There are things to think about, in particular what level of characterization to apply, as I discussed, you know, LCMS confirmation. If there are impurities of significant concern, you may have to go into further structural characterization. What level of the impurities are concerning enough to perform full characterization? Are you going to look at, you know, every impurity, or you're going to have a threshold, you know, any impurity is greater than 0.15 or 0.5 or 1%? These are discussions that need to be had, and evaluations that need to be made. In terms of the thresholds, there is no guidance. Oligonucleotides are excluded from ICH documents. There is a European Pharmacopoeia monographs for peptide, where a limit for peptides is provided as 0.5%, and given the similarities between peptide manufacturing and oligonucleotide manufacturing, it's a relatively good guidance to keep in mind. The Capaldi article on impurities suggested to use 1%, and it is an ongoing discussion within the community. But both the PHUR monograph and the Capaldi articles have not been vetted by regulators, and so it's something that is an ongoing discussion between pharmaceutical companies and regulators with respect to their particular drugs. The other aspect that is quite significant here is that these drugs are obtained by chemical synthesis, and purity is relatively poor as compared to, say, small molecules. The complex impurity profile means that it's difficult to separate the peaks from each other and from the main peaks. And so you know, in our case and in many other cases, there is value in using orthogonal HPLC methods. Otherwise, you won't be able to separate all of the impurities. In our particular case, we've argued that the complex impurity profile in the duplex and the fact that there is no chemical transformation between the single strand and the duplex means that it's easier to control the impurities really at the single strand stage. That requires the use of the same HPLC method for the single strand intermediates and for the duplex, but it allows us to specify most of the product related impurities in the single strand and only focus on degradants in the duplex. And that somewhat simplifies our work. It remains a relatively complicated aspect of this in terms of keeping an eye on so many impurities, especially, many of these impurities, in particular the N-1 or the N+1 impurities, are really not one impurity, but a family of impurities. Some are internal, some are terminal. And so it's somewhat difficult to keep them separated and consistently identified by HPLC, especially when sometimes they coelute, sometimes they don't, sometimes peaks are well separated, sometimes they're not. And so the better approach we thought is to use a specification that relies on groups of peak. This way the individual peaks don't have to be kind of quantitated individually. And so we applied this idea of using RRT ranges for groups of impurities in the single strand and the duplex. One of the important items is that regulators are still somewhat interested in the specific impurities within each RRT ranges, and so establishing a threshold and identifying the impurities in each RRT range is something relatively important to do. One of the problems with using RRT ranges is obviously retention time variability. It's very important to get very reliable peaks within an RRT range. Peaks cannot shift from one RRT range to another from sequence to sequence. And so that requires a level of normalization of the retention time. So some strategies have to be developed to allow these peaks to be consistently within the same RRT range from sequence to sequence. Otherwise, it becomes very difficult to specify impurity levels by RRT ranges. And so a certain need for that normalization exists. The other important factor here is as you go from single strands to duplex, you have to make sure that your degradants are well separated from the main peaks, that the degradants from one strand are not hiding under the other strand in particular, and so that they can be observed downstream of the single strands, and that's something that regulators have raised with us as well. So it's something that they are aware of. And so you have to ensure that your analytical methods are able to identify these degradants in the duplex as well as in the single strands. One item here that is pretty unique to this duplex type oligonucleotides is the fact that single strands are considered impurities. So in the duplex, if there is a little bit of sand strand or a little bit of anti-sand strand left over, it is not possible to argue that these are components of the drug substance and should not be considered any impurities. Regulators are looking at manufacturing capability in controlling them, and they are interested in understanding the impact of excess single strand on your duplex, so that you may have to conduct a toxicological evaluation of the single strands in the duplex if this is something that you cannot control to very low levels. And in our case, the excess single strand in the duplex is determined by a non-denaturing HPLC method. One final aspect of the purity profile is stereochemical purity. There is a lot of discussion now that there are a few techniques available to control phosphorothioate stereochemistry in particular. And so, you know, regulators are starting to look at this stereoisomer question a little bit more pointedly, you know, and starting to think of oligonucleotides in the way they think of small molecule drugs, and there is a potential that down the road stereoisomer specifications may be applied. In our case, this has not yet been the case. Stereoisomers are not considered impurities. There is an expectation that stereoisomer distribution be assessed. And there is an expectation that as you go through process changes in particular, you are able to show that stereoisomer distribution is not significantly impacted. If it is significantly impacted, then you may have to do an evaluation from a toxicological perspective. In our case, the stereoisomer distribution is evaluated on the single strands rather than on the duplex, where it becomes too complicated. And we've argued that stereoisomer distribution should not change downstream of the isolation of the single strands. So in summary, we've built some regulatory experience with oligonucleotides based on our experience with inclisiran. Regulators are still grappling with oligonucleotides. It's something relatively new and a regulatory experience being limited. But there is now more than a handful of drugs out there that are oligonucleotides. Regulators have taken note of the potential of oligonucleotides in treatment and the value and the benefit they can bring to patients. And so they are interested in this class of drugs. There is some acceptance that oligonucleotides cannot be treated as small molecules, and some understanding that their purity and impurity profile are somewhat more process related than they are than would be with small molecules. There is, however, an expectation that individual impurities be understood, that they're qualified and identified as they would in small molecules, that they are characterized and well understood, and that these impurity profiles are controlled adequately, and the idea that, you know, solid phase synthesis only gives material of limited purities is something they are starting to push back against. So you have to understand that your impurity profile needs to be controlled adequately. So this concludes my talk. In terms of acknowledgements, I wanted to thank Alnylam Pharmaceuticals. A lot of this work, and especially in the early stages, you know, came from the Alnylam school, and so we're indebted to them for this. I wanted to thank Marc Lemaitre, who was instrumental in holding our hand at the medicines company when we started to look at oligonucleotides, and his contribution is greatly valued. And I wanted to thank the Medco inclisiran team, the 3 people that you see there, and myself. This is the extent of the CMC team at the medicines company, and you know, it has been both a pleasure and a struggle to carry this drug through phase 2 and phase 3 and approval, or near approval, in you know, with such a limited team. And finally, I would like to thank you, the audience, for listening to my talk.